This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode 16 for January 16th, 2009. I'm Vincent Racaniello, and I'm in Switzerland with Jeremy Luban.
Hi, Vincent. Hello, everybody.
We're in Saanen, Switzerland, which is up in the Alps, right?
It's up in the mountains.
This is Jeremy's country. Well, we're here for a virology meeting, and what better place to do a TWiV than at a virology meeting? And today was the first session. Some interesting talks today. We have a couple of things to talk about on TWiV, the usual reader email, picks of the week. Are you enjoying this meeting?
Yeah, it's very interesting. It's a meeting that's been run essentially by clinicians in Switzerland, infectious disease docs, for this is the 19th meeting. They've had it every year, and it's a nice combination of basic science and clinical infectious diseases.
So have you been to one of these meetings previously?
I came once before. I was invited to speak once before. I gave a pretty basic talk. I don't know. It's the clinicians tend to be people who are connected to academic medicine or done research before, and so it makes for an interesting discussion.
So is this a course that the physicians can take for credit? Continuing medical education, as we say in the U.S.?
I'm pretty sure you can get credit, if you're a clinician, yes.
So it's a way of bringing together clinicians and basic scientists, basically, up in a beautiful place and just talk about science for 2 days, or virology. The name of the meeting is 19th Challenge in Virology. And today there were two sessions. We heard about HIV, elite HIV controllers.
Yep.
You remember we talked about that on
there was
previous
I seem to recall this once before.
A long time ago. And is this the same group? This is Bruce Walker, who was scheduled to speak. But was that his group who gave the talk today?
No, today was Phil Goulder, who was not on the paper we talked about before.
But he also works on the elite controllers.
So Bruce Walker was scheduled to speak and was unable to make it to the meeting. And according to the schedule, he was indeed going to talk about elite controllers and how, from the title, it looks like he was going to talk about how that information relates to thinking about developing an anti-HIV vaccine. Elite controllers are these remarkable people who spontaneously control the virus, we believe, via their immune function. Today there was a talk by Phil Goulder, who's Bruce Walker's in the U.S. and in Boston, and Phil is at Oxford in the U.K.
And Phil does controller work also, right?
Think so. I'm not actually sure.
I thought he spoke about it today and was also talking about HLA restrictions on susceptibility for HIV, so similar things. Then next was a talk on CNS complications of HIV, which is not an area I know very much about. And do you know anything about this? Or
I know it's very important to clinically. It's very important. I think it's fair to say it's an area that we really don't know a lot about. You can find evidence for HIV in the brain of people who are infected with it, but there's a lot of debate, disagreement, opinions about whether the virus is replicating there actively or how exactly it's getting there.
But the fact is, we have people with dementias and confusion, inability to function. What percent of people who have AIDS is this? Less than 10%?
I'm trying to remember. There were numbers reported in the talk. one of the points they were making is that I believe, is that if you look for it, that is, if you do formal testing, it's quite a significant percentage of people have some sort of impairment.
Yeah, it says that from my notes, 37 to 50% have mild cognitive disease, which is much higher than severe impairment. And one of his points was that if you treat them with the AIDS antiviral compounds, it doesn't always help because the dementia is not reversible.
Sometimes the drugs do in fact make a big difference when people have really evident dementia. Mm-hmm. There are times when it clearly helps. One of the problems, one of the topics that was discussed was a lot of the drugs, a lot of antibiotics, a lot of the drugs that are used to treat HIV, antivirals, don't have good access to the central nervous system, right, because of the blood-brain barrier.
Right. And
so there was some quite impressive clinical data shown today where large numbers of people were examined for the presence of virus in the blood, the presence of virus in the cerebral spinal fluid, which to obtain that you have to stick a needle in someone's lumbar region of their back and pull fluid out from.
Not easy to do.
It's
or
technically,
I should say, not easy to get people to consent to this, right?
Yeah, though I think they probably have some very committed people who are really eager to participate in these studies. Anyway, they measure drug levels. They looked for the presence of the virus and drug, and they showed that there are big differences between different antiviral agents in terms of how readily they get to the brain or to the CSF, and they showed that there are drugs where the inhibitory concentrations of the drug in the spinal fluid is about 30% what it is in the blood, and therefore it is conceivable you can achieve, by using high enough doses, you can achieve therapeutic doses in that part of the body.
One of the things I thought was interesting is that he mentioned that if you treat these patients with antiretrovirals, this increases their CD8 counts, and then that negatively affects the neural symptoms. So there's less improvement when the CD8 counts go up. What does it
Well, he said he was referring to, I don't know how generalizable that is. He said that there are case reports, there are isolated cases in the setting of giving the drug, giving certain drugs. Apparently, it was only with certain drugs.
I see. Well, that's interesting in itself too, right?
You may exacerbate the inflammation. I think their thinking was that in cases where you have relatively active inflammation in the brain, that there are times when you give the drug, you can precipitate worsening of neurologic symptoms, but I don't think that's a general. The idea that CD8s always go up when you start therapy wasn't familiar to me. I don't know.
Isolated. Well, the fellow who gave this talk was Scott Latender. Perhaps we'll have him on a TWiV in the future, and he can explain this.
Yeah,
because this is new to me, but I think it's very interesting. Yeah. The afternoon was spent on mainly respiratory viruses. We had a talk from Ian Lipkin, Columbia colleague, who is a virus discoverer, and it's absolutely amazing using techniques like PCR, mass tag sequencing, how you can isolate novel viruses from all kinds of specimens. And I'm hoping to get Ian tomorrow to TWiV with me. I don't know if I can corner him, but since he is at Columbia, maybe we can do that, because it's absolutely fascinating. He's got new viruses from all sorts of disorders. For example, he was the first to identify West Nile virus causing the outbreak in 1999. He was involved in identifying SARS. He's identified a new arenavirus in transplant patients, and recently he identified a virus that's killing honeybees and causing problems for the honey farmers. This is such fascinating stuff, and he is pulling out viruses for all of us to work on. So that will be very interesting. The other issue, there are two other things I just want to mention briefly from this afternoon. One talk on rhinoviruses from your colleagues here in Switzerland. That would be Caroline Taparel from Laurent Kaiser's lab.
Yeah, or
and
his
group.
In his group, yeah.
And they have identified a new group of rhinoviruses using molecular techniques. And these viruses are very difficult to grow, and that's probably why we haven't identified them before. But if you look for the nucleic acid, you can identify them. So that's going to be very interesting, of course. And then finally, we had some sessions on influenza virus, talking about how the virus drifts antigenically. Derek Smith from Cambridge was very good, and he's actually consented to join us on a future TWiV. We'll do that across the ocean. But he talked about how the virus drifts antigenically with time, how it spreads around the world, what are the kinds of patterns. Really good stuff. And then we had a session later in the afternoon called How Influenza Plays with Antiviral Agents. And this was actually a series of questions put up on the screen, and we all had clickers in our hands, and we would answer the question, and they would immediately put up the results of the question. And then Frederick Hayden from the University of Virginia would get up, and he would give us the information to try and figure out the correct answer. For example, talking about H5N1s and H1N1s and H3N2 influenza viruses, their resistance to various antiviral drugs, whether you should immunize older people, what do you do in immunosuppressed patients. And so here I can see where the clinicians who might see people with flu and have to decide how to treat them every day could learn from this experience. Did you like that?
Yeah, it was very enjoyable. In fact, I guess the way it was broken up, there were 3 or 4. It was broken up into 4 parts, maybe, and each part was planned by a particular clinician who offered a series of questions. Right. And then all of the people in the room got to test their mettle.
Right. I got most of them wrong.
Yeah, I probably got most of them wrong,
Which
too.
Means that I shouldn't be a clinician.
Well, it might just be that it's not really clear what the correct answer is.
For a lot of them, he was actually very circumspect.
Yeah.
But I think this is a great way to teach classes, to have us, instead of standing in front of people lecturing, have more interactive, where you put information on the screen and then you ask questions like this.
It's definitely more fun.
And if they don't get it, you go back and explain it until they get it. I would love to try that with a future course, so we'll see if we can do that.
It's especially effective if you have one of the world's experts in that exact area. Yes. It would be great to have, for example, a series of topics with the experts from each of those areas.
But that would be a great course.
That would be fantastic.
Yeah. Hayden today was absolutely knowledgeable about flu vaccination and antivirals, and much more than you or I could be.
Yeah.
Unfortunately, when you teach, that's what you're saddled with. You have to be an expert in a lot of things when you're not. So it makes it difficult. Well, tomorrow we have another day. We're going to hear about systems biology, which is a new way of studying virology. It's basically a way of looking at all aspects of the cell and what's happening during a virus infection.
That'll be, I guess, there's a talk from Emilio Talenti.
From Lausanne, right? Do you know him?
He was in New York at some point. He was at Einstein for medical school in his PhD that he did there, working with Bill Jacobs, if I remember correctly.
Tuberculosis.
And he I'm pretty sure he's originally from Spain, and then he started working as a clinician in Lausanne. And he's developed maybe I'm not sure how it originated, but he's developed an interest in human genetics and genetic determinants for disease progression, if you're HIV infected, or various properties, things that people are interested in when concerning HIV infection and disease. And so he has really committed himself over the past couple of years to looking for polymorphisms, single nucleotide polymorphisms that distinguish one person from another and confer susceptibility. He's had a couple of quite big studies published in the past year or two, and they've identified some loci with quite significant effects on viral load, for example. And the trick in most cases is to figure out what the mechanism is.
Sure.
And it's very hard to do that, to tie together a single nucleotide change with.
Well, at least we can do this. I mean, technology now makes it much easier to do this, to look at these single changes than it was 10 years ago. So that's good, and eventually we'll figure out how to get at the mechanisms as well. We also will have a talk on West Nile virus vaccine development by Michael Diamond, who's from Washington University, and he's actually agreed to do a TWiV in the future. So I'm cornering people here.
That should be exciting.
And then we have hemorrhagic fever, viral hemorrhagic fever talk, pathogenesis, epidemiology, drug development by Stefan Kunz from Lausanne. And he, I believe, trained at Scripps, right?
Yeah, I think he
Oldstone.
Just came back to. He's Swiss originally, and he just came back to Switzerland in the past year, I think.
And then tomorrow evening we have another question and answer to the experts, as we had today. This is going to be chikungunya, dengue, hemorrhagic fevers, rabies, vectors like mosquitoes, and West Nile. So that should be very interesting. And maybe I'll report to you, the TWiV listeners, next week when we do our next TWiV. So it's a great meeting. I'm glad I was invited to come and talk to them here. A couple of things we want to talk about today.
There was a talk today by this guy, Vincent somebody.
Oh, Vincent...
Racaniello.
Racaniello.
Yeah, no, that was that was a very interesting talk. So Vincent... He's kind of the break point in the history of polio. He defines the modern age of polio. I don't know how many of the listeners know this, but there was the first age of polio research, and then there was Vincent Racaniello who developed the first clones of the virus and the molecular techniques to study it. And in Vincent's talk, he actually started by describing some personal experiences he had. Personal encounters with a certain Professor Sabin. Vincent projected some actual letters that he had received, which were quite interesting, somewhat terrifying in how severely critical they were of a young scientist. Vincent contacted him I guess at the beginning of your career.
Yeah, at the very beginning of my independent lab, and when I came to Columbia, I first interacted.
And he basically accused you of one of three scenarios. Either you ignored what he wrote, or you didn't understand it, or you just thought it was a load of crap and didn't believe it.
Yes, those are his words. Yeah.
It's quite impressive.
I'll have to put those letters on the web. Someone actually asked me tonight to post them in their entirety, and I think I'll do that.
Those would be neat. But then he gave, then he went for the beef, and he talked about what's actually happening on the bench, he presented work from his PhD students, and that was really spectacular.
Thank you, Jeremy.
He showed how polio differs from other related viruses. They're all actually now called enteroviruses.
Polio and rhinos are both called enteroviruses, which is a bit counterintuitive because rhinoviruses do not infect.
One makes you sneeze and one makes you do something else, I guess.
Although I heard some things today which may suggest that some newer rhinos may infect the GI tract. Who knows? They're typically acid labile and temperature sensitive, so you wouldn't expect them to pass through the tract. But we'll see.
Anyway, I guess we're going to talk more about that in a second if I read your notes right. But just before we get to that, about what was really interesting about Vincent's talk was he showed how immediately after the virus, polio, enters a cell, it's like a chess game. It immediately starts attacking all of the sentinels that have been identified in the past few years that guard against viral infection. So there's a series of these viral detectors in the cell, proteins with names like RIG-I and MDA5 and IPS1 or Cardiff, whatever you call them. Then there's IRF3, and there's apoptotic pathways, there's interferon induction, there's interferon-induced genes, and polio is just wreaking havoc with
It's
all these
trashing
things.
them all, isn't it?
That's amazing.
You know, the way I view it is the innate response, which is the earliest response to a virus infection. It engages within minutes. It's so good that viruses would not exist unless they found ways around it. And this is just one example. But many other viruses, of course, trash this innate system. And pox virus is one of the more amazing ones. It trashes almost every step of this pathway. Now, we haven't actually talked at all on TWiV about innate responses, so this may be a little obscure, but
maybe I should interview you one day. So
one day you
talk
may.
about that.
We can talk about that. But yeah, the innate system responds early to virus infection. The end result is the production of interferons and cytokines, which end up clearing virus infections in the best scenario. But viruses have evolved to bypass them. Well, thank you for the brief summary, Jeremy. That was very
Sure,
nice.
that was much too brief. And if your students ever listened, their pictures were shown. They were discussed at some length
Yes,
during
they
the
were
talk.
discussed at some length. Yes.
But they should be happy. Their work was presented. And
3 things we want to talk about before getting to email. And probably we'll spend a lot of time on the second one. But the first, I just wanted to make a note of an article in the New York Times this past week, which was actually on the front page of the Science Times section, which is on Tuesdays. Is that correct?
yes, Tuesday is the Science Times Day.
And it's about a book written by Paul Offit. And TWiV listeners may remember last week we picked a book by Paul Offit called, or was it last week? Maybe the week before. In a recent TWiV, we picked a book by Paul Offit called The Cutter Incident. And in that he described the polio vaccine failure in the 50s. And now he has a book called Autism's False Prophets. And the title of the headline title in the Times article is Book is Rallying Resistance to the Anti-Vaccine Crusade. A new book defending vaccines written by a doctor infuriated at the claim that they cause autism is galvanizing a backlash against the anti-vaccine movement in the United States. And basically, as many of you probably know, many people in this country, and perhaps even outside the country, believe that some vaccines cause autism in their children. And there's quite a vehement and ferocious backlash by parents against immunization. You can find websites, you can find meetings where parents get up and say, we don't want our children vaccinated, we want a choice. Many scientists feel the opposite, that there's no evidence that vaccines of any kind cause autism. There have been a few papers which indicate that I've talked about in the past, and Offit's book is basically trying to lay out the evidence against that case. Now, I have not read the book. I just wanted to bring it to your attention. I will at some point read it. I've read some reviews that it's a bit biased. I personally feel that there isn't any scientific evidence that vaccines cause autism. But, this is a very complicated thing. And even if the vaccine is safe, it may be that in a certain child, under certain conditions, under certain genetic backgrounds, it does something that we don't understand at the moment. So we have limits to what we can do. But we do know that the vaccines prevent a lot of infectious diseases, and we have to be very circumspect about deciding not to take them. In a way, it's difficult for a book like this to come out and say this is all wrong. I think we need to have a dialogue which goes beyond this. And so we'll probably revisit this. I just wanted to point it out because I think it's very important. I've seen on the internet, on Twitter, I've seen a lot of backlash against this book already. People complaining that it's just to make money and it's another example of steamrolling vaccines, etc. And I really hate to see both sides of the argument turn to that sort of argument. I think we need to have a very calm and measured analysis and discussion of this, which I hope to have in future episodes. Do you know anything about this issue at all? Have you ever thought about or dealt with this in any way?
I've certainly thought about it a lot. I basically agree with what you said. I think clearly vaccines are something you don't just take them lightly. I think you take them for a reason. And I guess one issue is that we're in the United States and Europe where there is starting to be this growing anti-vaccine movement. We're really fortunate enough to be in a position where we can have this conversation. The vaccines most of the vaccines that are used routinely are there because if people don't take them, the consequences are dreadful. They're life-threatening, and there's no debate. Everyone sees it and experiences it. So polio is a good example. The people who grew up in the U.S. and during the years when the virus was rampant, everybody, I guess, knew people who were affected by it, who were crippled by it, who died from the viral infection. The development of the vaccine there itself may not have been a very pretty story. There's all kinds of historically, there's all kinds of stories about people fighting and cheating and doing dangerous things and all kinds of things. But in the end, the vaccine has been life-saving. And I don't think any parent who's ever seen a kid with polio would ever hesitate to give their kid a polio vaccine.
Well, having known Dr. Sabin a bit, I can tell you this was a guy that was just interested in preventing polio. He wasn't interested in making money. He never made money from the vaccine. He never patented it. He was just interested in saving kids. And I think a lot of people overlook that because vaccines are sold by companies that make money, and that taints them in a certain way because there's always the motive of profit, which is why I think vaccines should be a non-profit industry.
Yeah, or that the government should just produce them.
Exactly.
It's essential. It's essential part of the government function. But so that's polio, and a lot of people say, well, that's polio, but what about all of this other stuff? Well, if you've ever seen a kid with tetanus, again, you wouldn't hesitate to be sure that your kid was immunized against tetanus. It's got to be the most awful thing to see. I have seen the results of acute unimmunized infection with polio and tetanus. It's a memorable sight in both cases. Mumps kills people. It kills lots of people. These vaccines really are important. The problem with society and the vaccines is that if a large enough segment of the population decides they don't want to take the vaccine, it has consequences for everybody, and that's where the conflict arises.
Exactly.
So you say, okay, I don't want my kid to get this vaccine. If there's enough of you, you put other people at risk.
Okay. Immunization is not just about you, it's about everybody. It's a population issue, and that's the problem, that you can't just make individual decisions about this.
Yeah.
Now, I understand that parents get very upset when their children become ill at about the time that they get an immunization, but that's not cause for blaming the vaccine right there.
Unfortunately, it's not clear what to do about it, and
not at all.
This book apparently comes on strong, and may very well could do more harm than good. More harm in the sense that for people who believe that vaccines are useful.
Sure.
One clearly has to understand that people are afraid of vaccines for profound reasons. It's not a joke.
Yes, of course. I'm in no way making light of the whole issue. I just want our views are a certain way, and important to air them. All right, the second story we'd like to talk about, just a brief mention of the kind of immunization efforts that are underway in Pakistan. We'll put a link to this story. It's an article from the Daily Mail of Pakistan, or Islamabad. And it says a 3-day national polio eradication campaign will start from January 19th to immunize children under age of 5 years. Total of 30 million children will be vaccinated during the campaign. Campaign activities will be monitored by 500 monitors, a total of 43,000 vaccination team, 8,000 supervisors, and 1,200 zonal supervisors will participate. And the main focus is to give the live polio vaccine to kids living in high-risk areas. And the National Highway and Motorway Police will support the campaign by administering children at 58 toll plazas in the country. That means if you're driving through a toll plaza and they'll ask you, have you had a polio vaccine? You say, no, you're going to get one right at the toll plaza. And they don't have any choice. Now, Pakistan...
Can you imagine the delays on that road when somebody refuses?
Pakistan is one of 4 countries that still has wild polio, in addition to Nigeria, Afghanistan, and India.
Oh, that explains it. I was wondering why they were immunizing.
Yeah, there's still polio because of armed conflict has prevented them from immunizing in dangerous areas, particularly on the border with Afghanistan. The volunteers often can't get into the area, so here they're making a concerted effort. This is just an amazing effort. 30 million kids who are going to be immunized. 3 days. This is 3 days only. And this is the kind of effort that you need. And it's expensive, but WHO, of course, is paying for this with donations. So I just wanted to point that out because we don't do anything like this. Maybe we'll do it when measles is next, but it's just stupendous. All right, now the last story is actually an article in a room, sitting here in a room in Switzerland with Jeremy Luban, an AIDS expert. This paper is the one to do. It's called Direct Evidence of Extensive Diversity of HIV-1 in Kinshasa by 1960. This is a paper from Nature in October of 2008. I will just say one thing, Jeremy, and then we will hand it to you. As far as I recall, the oldest known HIV-1 sequence is from 1959, right? It's from a sample from Zaire. From that, people have tried to use sequences to argue that the virus came from monkeys to humans in the 20s or so. So what's the issue here? Why is this important? And what do we... First of all, where did this come from, this new sequence?
Despite all these different names, Zaire, Congo, they're different names for the same place. So we're talking about a region in Africa where there was a sample taken from that had been stored since 1959, and there was HIV sequence in that.
It was published in 1998.
98. Okay.
The paper is an African HIV-1 sequence from 1959, Nature 1998. So Zaire is what is today the Democratic Republic of the Congo. It's been renamed.
Yeah, it's been a very difficult place for people to live, to say the least, for the past century, and a lot of people think that has something to do with the development of the epidemic. It's one of the leading theories. In any case, what this paper is about is simply the identification of a sample. I think these were paraffin-embedded sections, tissue biopsies that had been stored in Kinshasa, or I guess at the time it was called Leopoldville.
Right.
From several eras ago, in fact. Several researchers were searching through pathology specimens, and they came across one specimen that was positive for HIV sequences. Sequences were pulled out, and it was done extremely carefully. It was done by different groups in different locations, and they compared results. And the end result was the identification of a second sample, the first being from 59. This is from 1960. The importance of this is that this sample from 1960 is significantly different from the sample in 1959. What that demonstrates is that HIV had diversified significantly within the human population by 1960, based on several different statistical methods that they go through in the paper, which I have no understanding of. They calculate a founding event, first virus that was transmitted to humans, to have taken place sometime in the beginning of the 1900s.
Earlier than previously.
Yeah. So I think the most recent possible date based on their statistics would be about 1930, but they're placing it most likely earlier than that, maybe in the teens or the first years of the 1900s. So right now, the main theory is that, and the evidence for this is very good, I have to say, and this I do understand because it's much more concrete, is that HIV was transferred to humans, or at least there's a record in the sequence of the HIV we have today of transfer to humans 3 times. And in each case, it was transferred to humans apparently from non-human primates, chimpanzees, that are present very close to Leopoldville in this area of the Congo and Cameroon. And
Is this from eating bushmeat?
well, nobody knows, but the theory is that somehow an infected chimp transmitted the virus to a person, and the most likely idea, most people think the most likely means would have been someone eating uncooked or incompletely cooked bushmeat.
Right.
So
What are the 3 times that roughly that we think this is transferred?
There were 2 transmission events that neither of which led to a huge pandemic. So you can find rare individuals who have viruses from those 2 transmission events, and those people with those viruses, the sequences that have been pulled out, those sequences have been linked to particular populations of chimps roughly in the same part of Africa. The vast majority of HIV-1 infected people in the world are derived from a 3rd lineage, a 3rd transmission event, which has also been tied to a particular geographically isolated group of chimps who have viruses with similar sequences, and therefore it's thought the transmission came from those chimps. And it was that 3rd transmission event, which is called the M group or main group of viruses, is the group of HIV-1 viruses that spread around the world. And within that M group, there are subgroups that are called clades, which comes from a Greek word, I think, for branches on trees or something like that. And they're named by letter, so there's clade A, B, C, D, etc. Each of these is a distinct branch that you can separate just based on the sequence into subgroups. But it's believed that all of these subgroups came from one single transmission event. The interesting thing about this one DNA sequence, or these sequences that were found in this one pathology specimen, is that they're closely related to, they're clearly within one clade, forget if it's A or D, and the sample from 1959 was in the other clade. So that indicates that already by 1960, the original virus that had been transmitted to humans had differentiated into these subgroups, which we see today. And that also gives statisticians a much better handle, 2 is much better than 1, on timing the original transmission event. So these 2 samples are theoretically derived from the same original transmission event, and it gives them 2 points to calculate with.
So we think by using the same approach, we can now look at other pathology samples from earlier years, perhaps. And
Perhaps.
Find others. How far back do we store? I mean, I know from the reconstruction of the 1918 influenza that we had pathology samples from back then, paraffin embedded in formaldehyde fixed sections. So in theory, we could go very far back for this, right?
Yeah, I think it's going to be much harder. 1918 virus was all over the world, and it was in major industrial centers all over the world, and so pathology centers in Germany and the United States had samples that they put in freezers and have been sitting frozen ever since. And then as well, there were people who died of the disease who were buried in Arctic or subarctic regions, who in one particular case were exhumed and analyzed. This is a pretty warm location, so any DNA that had been buried is certainly not going to be intact by now. I may be wrong, but it seems unlikely. And it seems much less likely that there would have been ongoing continuous refrigeration since the beginning of the century in this location. This could be the last of these early samples. Maybe there'll be a treasure trove found somewhere.
Well, in the article, they seem to think there'll be more.
There'll be more.
It says this opens the door to a detailed paleovirological investigation of the history of HIV-1. They have hope. I hope they're right, but
Yeah,
your points are valid, of course. It may be difficult.
Mm.
By the way, clade is from the ancient Greek meaning branch.
Ah.
Very good.
Wow.
Okay, you have a walking Wikipedia right here next to me. 2 questions. How long was the virus or related virus, SIV, I presume, in chimps? Do we have any idea?
I don't recall ever seeing anything that would explain that, or that would suggest a date there. It's believed that the chimps, the virus we now call HIV-1, we acquired from chimps, we believe. And in chimps, there is this virus that is, by sequence, is the same as the virus HIV-1 that's causing AIDS in people. It's believed that the virus that is in chimps, they acquired from other monkeys. Well, they're not monkeys. They acquired it from monkeys. Most people have the idea that humans are the only really violent primate, but chimps actually apparently can be quite violent. Apparently have wars, battles between different groups of chimps, and they also are carnivorous and will eat monkeys. It's believed that HIV-1 is derived from 2 other monkey species. That is, the same way perhaps someone in the Congo or Cameroon was hungry and killed a chimp and ate it and got HIV infected. A chimp ate a monkey and got infected with an SIV that had infected that monkey. But it's more than that. Apparently, the HIV-1 that is found in chimps is believed to be a recombinant, a combination of 2 different SIVs from 2 different monkeys. So at some point, there was a chimp that ate 2 different monkeys, each of which was infected with a different SIV. And somehow within that chimp, these 2 viruses recombined. So I guess it's could be possible that the chimp ate a monkey that was infected with 2 different. But the evidence suggests that the chimp ate 2 different monkeys. But this is just the tip of the iceberg. It seems that all of the non-human primates, if you look hard enough, you find viruses endemic in these wild populations.
Do gorillas have related viruses?
2 years ago, I think there was the isolation of an HIV-1 like virus from gorillas. Again, in very, very nearby.
A couple of weeks ago on TWiV, we discussed a paper where a lentivirus was found in the genome of a lemur. All right, so a virus related to SIV was found integrated into the lemur genome, and so this was one of the first times that these lentis had been shown to be integrated into germline cells, not just lymphocyte. Lemurs, of course, are on Madagascar, which is separated from Africa, and the last time it was near Africa was 14 million years ago. And in this paper, they speculated that maybe the lemurs got the virus from chimps at least 14 million years ago, maybe longer. And they said that this was evidence that the virus may in fact be older than we ever thought. So that would suggest that it's quite old, and maybe it's been in SIV, or SIV-like viruses have been in chimps for a long time. But it's not in their germline, right?
Yeah, no, I don't know of there being such a virus.
So it may be very old. So that brings the question then: why in the early 1900s did it? I mean, presumably did it jump into humans? Presumably humans ate meat before then. Why then? Of course, I know you can't answer that, but
the world was transformed in many ways over the course of the 1900s. So it seems likely to me that yes, HIV did infect people in the past, and for some reason it never took off into a pandemic as it did in the 1980s. I think it's probably just a question of ecology and of the conditions under which people lived and eventually lived over the course of the 1900s. So if people are living in small villages and are living in isolated villages, if there isn't communication over long distances, if people aren't traveling, if there aren't easy ways to travel, if you can't just drive a couple of 100 miles readily and go visit another city, if you have a social structure that's been existing for hundreds and hundreds of years and is relatively stable, maybe you won't get a pandemic. But if all of a sudden you have long-distance transport You have disruption of social structure and the form of forced slavery or urbanization or disruption of environments of all kinds. You have endless wars. You have young males in armies traveling back and forth. Everything changes.
And you mentioned this before with respect to the Congo. They've had a lot of unrest since the 1900s, I suppose.
Yeah. Kinshasa used to be called Leopoldville at the time. When the sample was taken, it was Leopoldville, and Leopold was from a country far away, Belgium. The Congo was a colony of Belgium, and there's a book that describes what terrible things happened in the Congo under the rule of King Leopold. There were forced labor camps, enormous forced labor camps. There were huge numbers of people who died, who were killed or died from starvation or malnutrition, who were forced to work to death.
What kind of work was involved? You mentioned before all this knowledge of the viruses and chimps and the different sources.
Well, it's a difficult problem. How do you get DNA sequences out of wild chimps that are protected or for other reasons?
You have to go where they live, right?
You have to go where they live, and you have to get samples somehow. And
virologists are not good at doing that sort of thing.
Well, there is one virologist, Beatrice Hahn, who seems to be quite good at it and has quite a number of papers analyzing sequences. She herself goes, and she has teams of people who are there, who go, and they've analyzed sequences from chimps in the region, maybe all the main groups of chimps. There are chimps here, chimps there. There's a river between these chimps that separates these two groups, and these chimps have sequences with one type, subtype virus, and these have another.
The chimps are infected, but they're fine.
So they seem to be asymptomatic as far as anyone can tell.
Typical zoonosis, right?
Yeah, I mean, maybe there would be a defect if the infected chimps were to be taken to the opera. They wouldn't appreciate it as much, to quote Harold Varmus, I think. But the way that they obtain the samples is particularly colorful. They don't shoot the chimps, dart them, knock them out, and draw their blood. They actually don't ever touch the chimps. They collect what the chimps leave. So they found that they could collect scat or that the chimps were leaving around, and that they were able to surprisingly obtain HIV sequences from these samples, also from urine. I remember Beatrice telling a story once at a meeting about how chimps, I guess like people, often like to urinate when they wake up in the morning, and so investigators would go out with collection devices and stand under trees where they might be able to catch them.
The chimps are up in the tree.
Apparently, yeah.
They wake up, stretch, and pee.
Right.
Typical.
So then they had to develop methods of identifying the chimps, and there were sequence markers for individuals. It's really...
Because they couldn't mark the chimps, obviously. They couldn't put a band around their leg. This is fabulous.
So they really have quite a detailed analysis of it.
And we have a lot to owe these people who have done that, because we've learned a lot about the origin of this virus, which is important. Yep. You can't do this for many viruses. Hantavirus is from mice. We were able to find the virus in mice. Now we're learning a lot about viruses in bats, particularly fruit bats. But the chimp is a rather stationary population. Aren't that many? And we know where a lot of them live. There are preserves also where they're protected. Yep. And so forth. Yep. Fascinating story, as usual.
Just a lot of interesting stuff out there.
Before we wrap it up, just let me read a few reader emails, and please jump in at any time. Richard from the UK writes, Dear TWiV team, first of all, many thanks for making this excellent podcast. I discovered it a few weeks ago and have been catching up with old episodes. I think you've pitched it at just the right level so that anyone with an interest in medicine, science can enjoy it. As a medical student, I found it a very good way of learning stuff while lying in bed. Perfect.
What's a medical student doing in bed?
He really shouldn't be, should he? I have a suggestion for Book of the Week. It's a short book called The Invisible Enemy, A Natural History of Viruses by Dorothy Crawford. The title pretty much says it all, but it's very well written and, like your podcast, pitched in such a way that anyone with an interest in viruses would find it interesting and informative. Experts like yourselves would probably not find anything new in it, but for those looking for a good introduction to the amazing world of viruses, I would definitely recommend this. So, I haven't read it. It's a recommendation from Richard. And we have an email from Everett. Frequently there is reference to women along the road to Kinshasa who, despite being exposed to HIV, presumably repeatedly since the beginning of the epidemic, have no trace of the virus. I suppose my first question is, are there such women who seem to have achieved a folklore-like status? And B, if they have no trace whatsoever of the virus, then are they controlling it in a way different from elite controllers? I don't know anything of prostitutes in Kinshasa that don't die eventually from AIDS. Do you?
This I had not heard of. It certainly might exist. I remember a while ago there were people in Kenya, I believe, who were highly exposed and apparently uninfected, and I seem to remember that they had remarkable IgA levels in their mucosa. Presumably in their mucosa. IgA is a type of immunoglobulin that gets secreted, and I don't remember exactly what happened. Maybe I can check that story out, follow it up, and maybe we can check out Kinshasa as well.
I looked around a bit for this and didn't find any. Mostly I found the numbers that over 90% of the prostitutes who work that road, which is a major highway for commercial traffic, die of AIDS. But we'll look into it some more, and the Kenyan story we'll look into.
Yes, I remember the Kenyan story was thought perhaps to be a different mechanism than what is being studied in elite controllers.
But all right, so Everett, we'll get back to you. And finally, is it possible that HERVs play a role here, human endogenous retroviruses? These are records of our previous battles with retroviruses, are they not? So then by default, does that mean there must be some segment of the population that is completely immune to this disease somewhere? Well, as far as I know, HERVs are retroviruses, not lentiviruses, right?
That's an interesting question. I don't know if Everett, this is from Everett, this comment? Perhaps he's referring to a specific study that came out, I don't know, 2 years ago, a year ago. It was in Close Pathogens, I think, from the lab of Doug Nixon. It's a very, very creative, very interesting story. The idea was HERVs are human endogenous retroviruses, and so we humans have them in our genome. And in general,
they're dead, right?
Most of the time, they're dead. There are very, very infrequently some that are potentially capable of making virus-like particles, making virus. Generally, these elements, even if the sequences are intact, they are regulated epigenetically. Their expression is shut off by host defense mechanisms that silence competitive elements like this, for example. I think there are several groups that had the idea that maybe HIV encodes factors that disrupt host defense mechanisms. And this may be, for example, HIV has a protein called VIF, which blocks a cellular antiviral protein called APOBEC. A couple of groups have come up with the idea that maybe when HIV infects a cell, when it brings viral proteins in, like VIF, that alter cellular antiviral factors, it might affect factors that affect the replication or expression of other viruses, like HERVs, like endogenous elements. And so there are reports that human cells that are infected with HIV upregulate the expression of human endogenous retroviruses. Doug Nixon comes from the same lineage as Phil Goulder, who we mentioned earlier, and has studied CTL responses, and that's been his bread and butter, anti-HIV CTL in HIV-infected people. And he wondered if there might be CTLs that recognize and kill cells that express HERV proteins. So normally the HERVs are suppressed. We don't develop an immune response against them. But maybe those cells that are infected with HIV turn off the silencing. These elements get upregulated. These proteins that aren't normally expressed get expressed and activate an immune response. And this paper in PLOS Pathogens actually provides some provocative evidence that such things might exist, and that these anti-HERV responses, since they're killing cells that are expressing HERVs, and the cells that express HERVs are expressing HERVs because they're HIV infected, indirectly, the immune system is killing the HIV infected cells. And that maybe people in whom these responses are strong actually have lower viral loads. I assume Everett was aware of this literature. Perhaps there are people who have particularly strong responses to HERVs, or they have particular HERV sequences that elicit strong immune responses, and therefore they have strong anti-HIV responses.
I think his point was that maybe these represented ancient infections with HIV, and as people survive and pass it on.
Oh, maybe I missed that.
But I think not, because they're not related to HIV. The HERVs are retroviruses, right? But that's a good, what you said is a good point. We'll put that Nixon paper in the show notes.
Maybe we should take it apart and talk about it.
Yeah, we can do that. Okay, one more email here. This is from Karen. Thanks for a great podcast. I am new to podcasts, but found yours when my PI, who also works on poliovirus, suggested it. After figuring out how easy they are to access, our whole lab listens to TWiV and a couple of others that you have recommended. I really like the background you give when you talk about specific viruses. For example, single-stranded versus double-stranded, host range, natural reservoir, recent outbreaks, etc. Two things I thought might be interesting to add to your list could be how the virus is causing the symptoms that you list, or whether it's the immune system like you mentioned with rhinovirus, and tricks the virus has evolved in the host to avoid detection, enter cells, or shut down the immune response. These sort of tricks are what first started fascinating me about viruses and got me interested in the field. Thanks again for a great show. You and all your guests so far have done a really fantastic job, and you make the show very interesting to listen to. Well, thanks very much for the comments. I think those are good points. We will try and focus on those issues, what causes disease, and how viruses avoid or evade various immune mechanisms. Certainly very interesting. We'd love to hear your comments. We appreciate your emails, and keep them coming. Okay, we'll wrap it up with our 3 picks of the week. First, the science blog of the week is called Eye on DNA, How Will It Change Your Life? This is a blog by Dr. Shen Lei in Singapore. And she writes, I'm passionate about social media and the potential it has to empower people to take charge of their personal health. The internet and genome technology combined are going to change our lives in ways we can't even begin to imagine. Medicine in the genome era will touch all of our lives, and I'm here to document its progress. DNA is our past, present, and future. We need to seize it, examine it, and know its power to make it work for us. How will it change your life? So this is a very interesting series of posts about, as she writes, how DNA is important for you, but more importantly, how the internet is going to make it easier for you to find out things about it. And I'm all for using the internet for science, so I like this blog a lot. And we'll put a link to that, of course. Our science podcast of the week is Persiflager's Infectious Disease Podcast. And I hope I pronounced the name correctly. This is an interesting one. I read the infectious disease literature, so you don't have to. That's the description. And basically he goes over every week or so, or every 2 weeks, all the ID literature, papers on viruses, bacteria, fungi, you name it. And he's got a lot of humor, good sense of humor. And it's, in fact, a disclaimer on his website that if you're offended by humor, me making fun of people, basically don't listen to this. It might not be for you. So we'll put a link in the show notes. I enjoy that a lot. You can find it on iTunes or...
His address is suggestive of the kind of humor.
www.pusware.com. In fact, sometimes he calls it the Infectious Disease Puscast instead of podcast. And finally, our science book of the week is one that was published in 1926, which I read when I was in high school. Made me want to work on bacteria. This is Microbe Hunters by Paul de Crieff. And basically he tells the stories of people like Lewinowicz, Spallanzani, Koch, Pasteur, Reed, and Ehrlich, and how they made their discoveries in a really engaging way. Great stories, real stuff, of course. These things actually happened. And so if you haven't read it, most of you may have already. If you haven't, be sure to pick it up. You can go to the TWiV bookstore on Amazon.
I guess I'll have to read it. I never
Ever read it? You should.
Read
it's very good. Very good book. Very short, and won't take you very long. TWiV is podcast of the week on another podcast called Net at Night, This is a podcast by Amber MacArthur and Leo Laporte, which I listen to religiously. It's one of the tech podcasts that got me interested in podcasting, and they picked TWiV for their podcast of the week. So that's great. We thank them for that recognition. And any of you who have come from Net at Night, welcome to TWiV, and we hope you enjoy it. Anything else you wanted to add, Jeremy?
Oh, well, Vincent, I was curious, do you have any plans for next Tuesday? That's January 20th. As far as science is concerned, it's a very important day. As far as Columbia University, it's a very important day because, as I understand it, that day will be the first day of the first Columbia graduates becoming a president of the United States.
Well, not many graduates of any university become presidents, right?
Yeah,
Only
I suppose
45.
that's true.
I don't know if they all graduated from college. But yes, Barack Obama graduated from Columbia University in 1983. It was the year after I came to Columbia.
Ah.
And
did you play basketball with him?
no, I'm not a good basketball player. He wouldn't have wanted to play with me. Although, judging by his kind character, he might have indulged me. But no, I didn't.
Did you know that there's a very famous virologist who seems to be advising him?
Yes, that would be Harold Varmus. We did speak about that a few episodes ago. You're not keeping up with your TWiV listening.
Oh, sorry, I didn't know you spoke about it.
Or actually, maybe we didn't mention it. Maybe I mentioned it on my blog. I don't recall. But yes, Harold is advising him, which is good.
And did you know that Harold Varmus has a connection with Columbia also?
Harold got his MD at Columbia.
That's right.
That's right.
It's a small world.
So we have a Columbia graduate becoming the president of the U.S. Do you know a president of Columbia, a former president of Columbia who became president of the U.S.?
Must be George Washington. George Washington Eisenhower?
Yes, that would be Dwight Eisenhower.
Dwight Eisenhower.
He was president of Columbia after the war and then went on to be elected president of the U.S. And
you had some interesting things on a blog once that he wrote.
Eisenhower was fabulous for science. He recognized that scientists were the real heroes that we needed to push. So he made this the first science advisor to the president. He began putting serious money into DARPA, which got the internet going. And he recognized... And
hence the podcast.
Exactly. We wouldn't be here without Dwight. And I will put a link to the post about that. He warned us about the military-industrial complex.
Ooh, that sounds like a good way to end this podcast.
And we didn't heed him. He said, we can't, scientists are going to be more and more dependent on the federal government because science is getting more and more complicated, and you have to let them keep their intellectual independence. And I think he was amazingly pressing.
Well, it's never too late to read what Eisenhower wrote.
Yes, you can find that speech where he mentioned the industrial complex. I'll put a link in the show notes to my blog post, and you can see that. But yes, Tuesday, President-elect Obama will become president, and we hope that it will be good for everyone, that he starts to fix things in the U.S. and helps get science back on its feet. You can reach me on the internet at www.twiv.tv. That's the website for TWiV. Or if you're on Twitter, PROFVRR is my Twitter handle. You have been listening to This Week in Virology. Jeremy, thanks for joining.
Thanks for inviting me.
I appreciate the time. I know you'd rather be at the bar drinking with scientists.
Of course.
This is a good cause, but tomorrow you can go back to the bar.
This was very enjoyable.
And we'll have you back again in the future. And thanks, everyone, for listening. See you next week.
Bye-bye.